welcome back to Equity Tutors and today's lesson, which is going to be on respiration. So we're going to dive into how our cells essentially generate energy from glucose. And now we're going to break that down into three crucial stages: glycolysis, the link reaction, and the Krebs cycle. So first, let's talk about fundamentally what is respiration. How do we define respiration? So respiration is the process by which our bodies break down glucose to release energy. And energy is generated in the form of a molecule called ATP. And ATP is then able to power processes in the body, things like muscle contraction, cell division. Every essential function of our body requires energy, which is in the form of ATP. So respiration is a really fundamental process to the body for us to function. So when I talk about these three steps of respiration, I'm mostly talking about aerobic respiration. This means that it is dependent on oxygen. Anaerobic respiration is independent of oxygen. So aerobic respiration takes place in multiple stages, as I just mentioned. We have glycolysis, link reaction, Krebs cycle, and then following Krebs cycle, oxidative phosphorylation, which I do not expect we'll cover in this lesson, but it'll be in the part 2 of this respiration lesson. So during aerobic respiration, glucose is effectively burned out inside our body with oxygen to produce carbon dioxide, water, and, as I mentioned, energy in the form of ATP. So the overall reaction is glucose plus oxygen is carbon dioxide and water. And I've also included here the chemical formulas for each of those, and you can also see the number of each of those molecules that goes into. So for every one molecule of glucose, there are 6 oxygens required, and that makes 6 molecules of carbon dioxide and 6 molecules of water, or H2O. So let's start first with the first step in aerobic respiration, which is glycolysis. So this occurs in the cytoplasm, outside of the mitochondria, and glycolysis is essentially the part where the first part we start to break down glucose. So glycolysis, lysis meaning to break down, and glyco referring to glucose. So we start with glucose, which is a 6-carbon molecule, and we're going to turn it into 2 3-carbon pieces called pyruvate. So breaking it from a 6-carbon to 2 lots of 3-carbon molecules. And this stage is independent of oxygen, so it's happening at all times in your cells. When we go on to learn about anaerobic respiration, that occurs actually after glycolysis. So let's go through step by step. Hopefully you are looking at the diagram I have on screen. So as I mentioned, that the input to glycolysis is a 6-carbon glucose molecule, and the output is 2 pyruvates, which are 2 3-carbon molecules. But also in between, we have 2 triose phosphates. So glucose firstly gets broken down into 2 triose phosphate, and that is an ATP-dependent reaction. So the ATP gets reduced to become an ADP, so it loses a phosphate group, and that phosphate group is then consumed by these 2 triose phosphates, and then you get a second reaction from 2 triose phosphates to 2 pyruvate molecules. And again, that requires 4 ADPs to be made into 4 ATPs. And in addition to that, you also get reduction of NAD to NADH. So let's go over it step by step. Glucose becomes phosphorylated using the phosphate groups from 2 molecules of ATP. So ATP is hydrolyzed into ADP and an inorganic phosphate. So that's the first step of going from glucose to triose phosphate. The ATP is hydrolyzed, releases an inorganic phosphate. This forms a molecule with the glucose, which is unstable, and causes it to break down into 2 3-carbon molecules called triose phosphate. So the triose phosphate is kind of an intermediate step, but it's pretty unstable. And what then happens is that the hydrogen is removed, and then that's what forms the pyruvate. When the hydrogen removed, it's actually transferred to a coenzyme, which you'll be hearing a lot about in this lesson today, called NAD, to its reduced form called NADH. The removal of the hydrogen from the triose phosphate oxidizes it, and then the reduced NAD is used in the last stage of aerobic respiration, which, as I said, we'll get to either late in this lesson or in next week's lesson. But we are now going to follow what happens to the two pyruvate molecules. And that is going to go into the next step, which is called the link reaction. But let's look firstly at the currency here. So how many glucose molecules do we have going in? How many ATPs, ADPs, etc.? So we have conversion of a glucose into 2 triose phosphates to 2 pyruvates. That produces 4 molecules of ATP. So remember though that we actually used 2 molecules in the first step to phosphorylate the glucose. So there's a net gain of 2 ATP molecules in glycolysis. We use 2, but we make 4. Okay, so the next act, the second act in aerobic respiration, is the link reaction. And this is happening inside the mitochondria. And you want to make note where each of these stages are occurring because they are very common 1-point answer exam questions. So this is the link reaction because it connects glycolysis to what's going to be the big show, which is the Krebs cycle. So in this reaction, pyruvate, remember we have 2 pyruvates that we made from glycolysis. They are 3 carbon molecules. They go through another transformation to be able to become part of the main production process before entering the Krebs cycle. So during this makeover, pyruvate gets rid of one of the carbon atoms, which becomes a carbon dioxide, which we then breathe out. Again, relate this back to the main respiration equation that you are familiar with. You know that we produce carbon dioxide, and then the other 2 carbons combine with a molecule called coenzyme A, forming acetyl-CoA. And acetyl-CoA is now ready for the Krebs cycle. So that is the molecule that's going to enter into the Krebs cycle. I know this is a lot of names to remember. It seems very much like a memory test at times. I completely agree. But what is good about sometimes having a memory test is that you are less likely to get really challenging questions. And once you learn it and once you know it and you've memorized it essentially, and this question comes up on an exam, there's no nuance in the answer. It is very black and white, and if you've learned and you've put the hours in to memorize it, you are going to get all of these marks. So let's just go over that very quickly. The link reaction is basically going to transform the 2 pyruvate molecules that we got from glycolysis into an acetyl-CoA, which has 2 carbons in order to enter the Krebs cycle. This is occurring in the mitochondria. Actually, more specifically, it's occurring in the mitochondrial matrix. It does not produce any energy in the form of ATP, but it does produce a reduced NAD and acetyl-CoA, as I just said, that 2 carbon molecule. Reduced NAD will be used up again in the oxidative phosphorylation, while the acetyl-CoA is going to be used in the next stage. So let's look at it step by step. Again, I hope you are looking at a screen and you can see the equation that I have laid out. So the first step is breaking down of the pyruvate to form an acetate molecule. That's a removal of a carbon dioxide. You get a release of it's a removal of a carbon atom, and then you get a release of a carbon dioxide. And then there's also an addition of a hydrogen to reduce the NAD to form an NADH. From the acetate molecule, that then interacts with a coenzyme A to form acetyl-CoA. And then once again, I want to take note of what has this part of respiration produced and used up. So for 1 glucose molecule, we get 2 pyruvates. So for every glucose, the link reaction is happening twice, one for each pyruvate. So this means that each molecule of glucose produces 2 acetyl-CoAs along with 2 carbon dioxide and 2 reduced NADs. Okay, so next up we have the Krebs cycle. So the Krebs cycle I think can be a little bit scary. So I'm just going to give you a vague overview to begin with, and then I'll go deeper and deeper into detail. So essentially what is meant by the Krebs cycle, it is a continuous loop of carbon-based molecules getting the carbons added and taken away. And through this loop and the energy and interactions between the molecules of adding and removing carbons, we get energy produced in the form of ATP. Now we know from our previous explanation on the link reaction that we have a 2 carbon molecule called acetyl-CoA that is going to enter into the Krebs cycle. And immediately upon entering the Krebs cycle, the acetyl-CoA will combine with a 4 carbon molecule to form a 6 carbon molecule. It'll then go from a 6 carbon molecule to a 5 carbon molecule, back to a 4 carbon molecule, and that 4 carbon molecule will combine with a second acetyl-CoA to form a 6 carbon molecule, and the cycle goes around and around. And the Krebs cycle is the most important process for our bodies to produce energy. So it's going to produce loads of ATP, but it's also going to produce a bunch of other stuff, things like carbon dioxide, reduced NAD or NADHs, which we also need to make note of, and make note of where between which 6, 5 or 4 carbon molecules that those gains and losses are occurring. So acetyl-CoA is going to combine with a 4 carbon molecule called oxaloacetate. That then forms a 6 carbon molecule called citrate. So now from the 6 carbon molecule, we're going to start to lose carbons. Every time we lose a carbon, that gets released in the form of CO2. So we breathe out the CO2. Between the 6 carbon citrate and the next 5 carbon molecule, we do lose a CO2, and we also reduce an NAD in the form of NADH, which gives us a 5 carbon molecule. And then in the next stage, there's a lot of things that happen between the 5 carbon molecule and the 4 carbon molecule, which we now know is called oxaloacetate. So the first one, as you'd expect, is you lose carbon in the form of carbon dioxide. And then the same thing again, we get a reduced NAD, so we get an NADH produced. The next thing that happens is that we get a reduction, an addition of a phosphate group to an ATP to form ADP. And then 2 more steps. First, we're introducing a new molecule called FAD. This is similar to NAD. It gets reduced, meaning it adds a hydrogen. So you get a reduced FAD or an FADH, and then you get another round of reduction of NAD. So you go from NAD to reduced NAD or NADH. So all in all, between the 5 carbon molecule and the 4 carbon molecule, first one, easy to remember, you're going to lose a carbon in the form of carbon dioxide. You get a production of an ATP molecule, you get a reduction of NAD twice, and then we also get a reduction of a new molecule called FAD. And then from there, we're back to the 4 carbon molecule, which reforms with the acetyl-CoA to form the 6 carbon molecule citrate, and the whole thing repeats. The reduced NAD and reduced FAD will be used up in oxidative phosphorylation. And then I also wanted to introduce you to 2 other terms, which are decarboxylation and dehydrogenation. So these are the fancy official terms we use when we remove a carbon or remove a hydrogen from any molecule. So I can say that the 5 carbon molecule was decarboxylized to form the 4-carbon molecule. So let's go over it one more time, and then I'm going to go through each step one by one again. So the Krebs cycle is essentially a series of reactions which generate reduced NAD and a similar molecule called reduced FAD, which are required for oxidative phosphorylation. We're also producing ATP. Acetyl-CoA from the link reaction reacts with a 4-carbon molecule called oxaloacetate. The coenzyme A portion of the acetyl-CoA is removed and returns the link reaction to be reused. This forms a 6-carbon molecule called citrate, and sometimes the Krebs cycle can be known as the citric acid cycle. Carbon and hydrogen are removed from the citrate, so it's decarboxylated, dehydrogenated, forming a carbon dioxide and a reduced NAD. The citrate is then converted to a 5-carbon molecule. Decarboxylation and dehydrogenation occur once more, which converts the 5-carbon compound into a 4-carbon molecule, the oxaloacetate, which we started with. And in this process also, we get a molecule of ATP, 2 molecules of NAD, 1 molecule of FAD, and a carbon dioxide. And this cycle is going to take place twice for each glucose molecule that's respired aerobically, and that is because with each glucose molecule we get 2 pyruvates, which means 2 acetyl-CoAs, which means 2 Krebs cycles per 1 glucose molecule. So let's go back, and now that we understand the whole process, I know this is a lot of information, but go back and look at each step one by one again. Just a quick overview. So starting with glycolysis, we have 1 glucose molecule which gets broken down into 2 triose phosphate 3-carbon molecule products, and that is because it gets phosphorylated by the ATP, forms an unstable molecule in triose phosphate, and that then immediately gets converted to a pyruvate via reduction of NAD to form an NADH molecule, and then we produce 4 ATP molecules. So we've gone from glucose to pyruvate, making 2 net ATPs and 2 NADHs. The ATPs are going to go away and get used for energy for all our cellular functions. The NADH is going to be used in oxidative phosphorylation, but where we are going today is following the pyruvate into the link reaction. And that is all occurring within the cytoplasm. So the next we're going to go into the link reaction. Again, we're following the pyruvates. This occurs in the mitochondrial matrix and is going to convert the pyruvate into acetyl-CoA. Again, this is occurring via 2 different steps. Firstly, the pyruvate gets converted into acetate. So this is again losing a carbon atom and generating a CO2. And it also is dehydroxylated, so we get a reduced NAD to an NADH. This intermediate molecule is called acetate. It combines with coenzyme A to form a acetyl-CoA, and this is what is going to take us into the Krebs cycle. So remember, the acetyl-CoA now has 2 carbons, and the acetyl-CoA enters the Krebs cycle, combines with a 4-carbon molecule called oxaloacetate, releases that coenzyme A that goes back into the link reaction, and we now have a 4-carbon molecule called citrate. That gets reduced to a 5-carbon molecule by decarboxylation, dehydrogenation. So that's releasing a CO2 and again another reduced NADH, a reduced NAD, or otherwise known as NADH. To get a 5-carbon molecule, the same happens between 5-carbon molecule and a 4-carbon molecule. We get a release of CO2, an NADH molecule. We also get another ATP, and then an additional NADH and a similar molecule to NADH called FADH, and that takes us back to the 4-carbon molecule, which is then there waiting for the next acetyl-CoA to enter the Krebs cycle. So I think I'm going to leave the lesson there today, just because this is a lot of information. It requires you to go over it multiple times in order to remember everything. But just to recap, we covered glycolysis, the link reaction, the Krebs cycle. The next step is going to be oxidative phosphorylation, where we're going to use up all those reduced NADs. And then next week we'll also look at anaerobic respiration, and then how in the lab we can measure respiration as well. So remember to like and subscribe if you enjoyed this lesson, and please leave any comments or questions that you have below if anything isn't clear and you need any further explanation. Bye!
Bye!